Well, hey, AP Bio students, Mrs. Shelton here. I am going to talk to you on this screencast about chapter 23. And actually, what you're seeing here, this is one of my favorite animals from being an AP Bio teacher. This is an axolotl, and we will talk about him. This is my favorite picture. This used to be the picture that was used in the textbook. I'll show you the current book, the picture that's used in the book, but this is a cute little salamander that actually displays kind of an interesting genetic change that we will address as far as part of our broad patterns of evolution, which is what chapter 23 is all about. So things that you guys should have at this point in time, you should have your chapter 23 notes that's available for you on Schoology, and then also your textbook if you want to follow along with some of the pictures that I'm providing for you. But we are going to be just talking about some of these major events. So again, these broad patterns of evolution. This is our last chapter for our natural selection unit. And obviously, when we talk about some of these broad patterns, these macroevolution events, so these major changes that we can see that get outlined from the fossil record, we have to understand that most of our evidence of all of these broad patterns of change comes from fossils. Now, what we're seeing here is we are actually seeing whale bones in the desert. But what this is an inland ocean. We can actually see some of the evidence of the water breaking it down. But we need to understand our fossil record and what fossils actually tell us. Now, I copied for you actually two really important quotes that we have discussed. I mean, we look at some of these fossils here of different whale bones. We've got vertebrae and we've got ribs from these. But the two quotes, they actually seem really contradictory. And I need you guys to make sure that you understand why they don't really contradict. So the first quote that I have on your notes for you is the fossil record is an incomplete chronicle of evolutionary change. So understand why it's incomplete. I mean, let's think about just that process of forming a fossil and how many species actually do leave behind any fossil evidence for us to discover. Then the second quote, that again seems to contradict the first one, is the fossil record is a remarkably detailed account of biological change over the vast scale of geologic time. So again, incomplete, but it really does show us this complete picture. Because again, that improbability of fossilization, but now we take the course of these billions of years of this accumulation of fossils, we really do get a good sense of these broad patterns of evolution and this adaptive radiation that has happened over the course of life on this particular planet. So let's detail wise in this particular section in 23.1, when you read 23.1, the most important piece of 23.1 that you get through is I want to just kind of look at some of the cool pictures and this the adaptive radiation, these changes within the fossil record that we see. You do not need to be held accountable for any of these particular terms or any of these particular major events, but the absolute important part of this particular section is understanding how we understand how old fossils are. And most importantly, when we do find fossils, it is radiometric dating. And when we say radiometric dating, what we are using is we are basing that on the decay of radioactive isotopes. So remember that a radioactive isotope, when we talk about an isotope, isotopes have an unusual number of neutrons in the nucleus of the atom, which makes the nucleus unstable. So it's radioactive. And so what happens when we do talk about radioactive isotopes is isotopes will decay. And when they decay, that means the nucleus will break down. And when that nucleus breaks down, either through alpha or beta radiation being released, what will happen is that atom will change identity. Because remember, the identity of an atom is based on number of protons. So we can actually look at what's the new, what is the new amount versus how much is the old amount by calculating the half-lives. Now, this is a calculation you're going to need to do when there's several practice problems for you to be able to determine the age of a fossil based on a known half-life. So when we do talk about half-lives, every radioactive isotope has a known half-life. It's the set known amount of time it's going to take for half of the parent isotope to break down and decay. So what we can actually see here, and what you would need to know, is how many half-lives has this particular isotope have. So what we understand here is if I had my whole of my radioactive isotope, at the end of one half-life, I have half of the mass of that remaining, and the other half now is a different atom. Now, another half-life, I now have a fourth of that material left after two half-lives. And so if I only have a fourth of the radioactive isotope remaining, I know that I've gone through two half-lives because here I have half. What's half of half? Well, half of half is 1/4. So 1/4 tells me that I've gone through two half-lives. Now, if I've gone through three half-lives, I have 1/8 of the original isotope that is remaining. Because again, another half-life, I have half of a fourth. What's half of a fourth is 1/8. So that tells me I have three half-lives. And then if I have 1/16, well, what's half of 1/8? Half of 1/8 is 1/16. That tells me I've had four half-lives. So how you would use this is if I told you that I had found a particular isotope and that particular isotope had a half-life of 1 million years. Just say this. So if I had, that half of that isotope had a half-life of 1 million years, and I have determined I have 1/8 of the parental isotope left, well, that tells me I've gone through three half-lives and that this particular fossil is 3 million years old. So we would always multiply how much we use our fraction of the isotope that's remaining to determine how many half-lives it's gone through, and then we multiply that by the known half-life of that particular isotope that we're using. So some of those practice problems, we will address those in a little bit when we have some video conferencing. Now, in 23.2, what you need to be addressing in 23.2 is understanding, and so the title of that particular section is The Rise and Fall of Groups of Organisms Reflect Differences in Speciation and Extinction Rates. So one of the concepts that's addressed here is just understanding plate tectonics a little bit. So when we talk about plate tectonics, we understand that the Earth's crust is actually floating on this mantle layer, and our plates, these are movable, and these are moving plates that we can measure on a regular basis on how much they're moving away from each other or towards each other. And with this, that result that's happening with plate tectonics, these plates moving that we refer to as continental drift, what is happening here is, we're going to have changes in the separation, we're going to have changes in climate, we're going to have changes in all sorts of things that are happening as a result over the course of hundreds of thousands to millions of years. Now, you all know Pangea. So Pangea was that one big landmass that existed. Google, when was Pangea? How long ago was that? To now where we are here and understand just some of those differences that accumulate as far as those consequences that happen as these masses shift and move with taking on them all the passengers, all the organisms that are inhabiting that terrestrial land. Now, when we do talk about broad patterns of evolution, we cannot ignore mass extinctions. When we talk about the history of life on this planet, it's really important to know that we have had 5 what are considered mass extinctions that have existed. When we say it's a mass extinction means that at least 50% of all marine life has gone extinct, no longer exists. This over here, so this is the total extinction rate. Over here on this y-axis is the number of extant families, so those that existed. And then when we see these spikes, the extinction rate, here is the first mass extinction. Here is a second mass extinction that happened here. Here's a third one that happened, a fourth one, and then also a fifth mass extinction. Again, being marked with at least 50% of marine life that go extinct during that particular period of time. Now, as You don't have to know each of these five, but you should be familiar with two of them, and you should be familiar with what actually caused that particular extinction. So what you have here in this huge, huge spike here, this Permian mass extinction that happened here. So in this Permian mass extinction, 252 million years ago, um, approximately 96% of marine life went extinct during this particular time period. What caused that? Volcanic activity. High levels of volcanic activity actually increase carbon dioxide levels in our atmosphere, caused massive amounts of ocean acidification, which led to mass extinctions occurring. Now, a second mass extinction that you guys should know about is you should know about the Cretaceous mass extinction that's happening here. So this Cretaceous mass extinction, this was our most recent mass extinction of 66 million years ago. And so during this Cretaceous mass extinction, with this, there's evidence that shows that this was caused by an asteroid. And so the asteroid or meteor fell in the ocean, leaving lots and lots of different dust, and accumulation of material in the atmosphere that changed the weather patterns that led to a mass extinction as well. So I'm telling you these not so you memorize these particular mass extinctions, is you recognize some of the different causes of mass extinctions. And it usually has to do with massive changes in climate, which then, if your climate is changing, then with that, you are also changing now that photosynthesis, that productivity that's happening in an ecosystem, which now limits your resources for energy. So one of the questions that needs to be addressed and asked is, are we currently under a sixth mass extinction? Um, that's something that we don't know. I mean, extinctions don't happen over the course of a couple hundred years. These mass extinctions, and it's only time will tell, is currently is human activity actually leading to mass extinction. Oh, cat trying to help me out. Um, Now, when we do talk about mass extinctions, though, we actually have to talk a little bit about the consequences of mass extinction. Because when a mass extinction happens, like when the dinosaurs went extinct, that's what we want to discuss. Those large energy-consuming reptiles that were on our planet, well, those don't exist anymore. But that leads to more niches and more resources for other organisms to be able to evolve and radiate from there. So big consequence of mass extinction, we are a consequence of mass extinction. If those mass extinctions hadn't happened, humans wouldn't have evolved. There wouldn't have been a niche for that type of radiation to occur. So keep that in mind as far as what ends up happening. Now, when I did say that mass extinctions lead to adaptive radiations, when we talk about an adaptive radiation, we are talking about this one common ancestor that now is diverging into other ancestors. And then here's our broad patterns here. I mean, we're just talking here about monotremes to marsupials to eutherians. But there was an ancestral mammal from this radiation in order for this to occur. If we're talking about a worldwide radiation, I mean, this is again on a global scale, not just within one particular isolated area. And we can look at some regional adaptive radiations. Here it's talking about a native species of plant that actually radiated into all of these other species over the course of a few million years on the Hawaiian Islands. So that would be what a regional large scale evolutionary radiation would look like too. So just kind of keeping those in mind. Now, chapter 23.3, this particular section in 23.3 is actually the title of that particular section. Major changes in body form can result in changes in the sequence and regulation of developmental genes. So when we talk about developmental genes, and so remember that these changes, whether they're mutation, whether it's change in regulating just gene activity, whether it is now gene duplication, things like that, we need to understand some of these terms that are affecting some of these small changes in genes can actually have some pretty huge morphological changes in organisms. So one of the terms that I need you guys to know is the term heterochrony. So that term heterochrony, what it's referring to is the fact that, like, just for example, like bones in our body all have different timing of when they stop growing. So, for example, I mean, the bones in your skull they stop growing at a different time period than, say, your femur, your leg bones do. So, and that's all controlled by genes as far as what controls when certain bones start and stop growing and when they mature. Now, for humans, so this heterochrony is that fact, that timing of development and growth is all controlled by genes. So now you alter those particular genes, now you're going to have changes in shape. And so imagine what would we look like if our bones in our skull continued to grow at the same rate as the rest of our bones in our appendages, so in our arms and our legs. You would look like Jack in the Box, is what you would look like. You would have a big old skull, is what would happen. Now, here is Mr. Oxalotl. Now, Oxalotl, again, is a salamander, and this particular salamander, when salamanders are in development, what actually happens is they have gills that are supposed to be turned inward. So they start with external gills, and then those gills, this is for oxygen exchange in the water, are supposed to turn inward and have these inward gills. Well, during that fetal development, from that juvenile state to the adult stage of turning inward, well, there's genes that control that change and that transition. So for oxalotl, well, oxalotl, that particular mutation occurred where those, what you're seeing here, it kind of looks like a little happy hairdo going on here. These are gills that didn't turn inward. These are external gills, is what these are. Now, for him, this particular species, this works because he's underwater all the time. And so having external gills ended up being a mutation that worked just fine for that particular salamander species. This event, when we actually have these developmental transitions that, again, are all controlled by genes, when we have mutations in those developmental transitions, that's called paedomorphosis. So that term paedomorphosis is, again, those developmental genes that are turning off or turning on. In humans, during embryonic development, we have gill slits, we have tailbones. So there has to be that paedomorphosis, those changes in those features that also change as well. Now, we've talked about our homeotic genes or Hox genes. And so these homeotic genes or Hox genes, you have a mutation in any of these. Well, remember, these are controlling body arrangement and spatial development. So there's a few genes that are really important for these morphologies. So what I just gave you, 3 different examples of how just some small changes in genes can actually have huge morphological differences and morphological change if there's change in that genetic makeup or in that control of gene expression. So heterochrony, that timing when growth is occurring, paedomorphosis, that transition from an embryo to a juvenile state to an adult state. So there's genes that control those. Or we also have those homeotic genes or Hox genes would be the specific example. Those are for spatial arrangements and controlling the spatial arrangements where different limbs or body segments actually appear as well. So you have changes in those, you get some pretty huge morphological differences. Now, when we talk about, actually, let's go back here just a second. Because, again, with this topic of developmental genes, and I gave you 3 examples here. There is a term that you guys will go, Mr. Oxalotl, because he's so cute. So one of the things that that you've probably heard of as we talk about these broad patterns of evolution is the Cambrian explosion. And with the Cambrian explosion, that actually was about 1 million years. So it was 1 million years in animals where there was a series of rapid change within animals within that animal kingdom. Now, that was about 535 million years ago when the Cambrian explosion occurred. But this rapid diversification of animals was actually caused because, and what DNA evidence is really showing us is the fact that there was a lot of gene duplications in these regulatory genes. So like what I just explained to you, so heterochrony, paedomorphosis, homeotic genes. Gene duplications, these changes in some small mutations that were occurring, and then also just in that gene regulation of some of these morphological control genes happening. So this is kind of those evidences when we say change in gene sequence or change in gene regulation. It's in these developmental genes that are controlling those timings and those transitions that they really believe led to this Cambrian explosion and all of these different morphological shifts within a pretty rapid time period of about a million years. That was the Cambrian explosion. But again, sexually reproducing organisms and the animal kingdom is where that exists. Now, the last topic that we want to address is actually 23.4. And with 23.4, the title of that section is Evolution is not goal-oriented. There is no master plan for evolution. Evolution is a process, and it's a process of natural selection. There's a lot of just, we build on top of existing modifications. So even this quote that I included for you from Francois Jacob, or maybe it's Francois Jacob, I don't know. Evolution is a process in which new forms arise by a modification of existing structures or existing developmental genes. So evolution it's a process of building that descent with modification. So when we look at like things like evolutionary novelties, your eyes are considered an evolutionary novelty. Now, evolutionary novelties, they didn't just bam, here's an eye. They started out as, here's a few cells that actually these respond to light. So, and they communicate that response to light to nerve cells. So that's the beginning of it. And then from there, well, now there's an eye cup that maybe evolves, a few 100,000 years later in another related species. And then maybe there's, this evolution. It's built upon and built upon and built upon. When we end up having these huge complex structures, we again have to recognize that this evolutionary process, we build upon it. It's a process. And again, we don't just have these evolutionary novelties that just appear from nowhere. Now, there are some things that do happen too, though, where, again, as an example of evolution not being goal oriented, there's not a master plan that happens. And there's not a picture for this, but what we should be aware of, like your ears. So here's an eye, obviously, but your ears, when we talk about your ears, I mean, this is not for hearing, this is just for collecting sound and pushing it back through your eardrum. And so those sound waves are vibrating your eardrum. And then after your eardrum, you actually have three bones that are your malleus, your incus, and your stapes. So those are referred to as your inner ear ossicles, these three bones that actually amplify those sound waves that are coming through from me right now. So those auditory ossicles actually did not actually evolve as bones for hearing. They're what are referred to as an exaptation. So that's that last term on your notes that you need to know is those inner ear ossicle bones, their exaptations, they actually evolved in another organism, part of the jaw hinge bone. And so those bones were part of the jaw structure, but again, building on top of it as evidence there's no goal oriented, they actually became beneficial in amplifying sound. And so that's where that evolutionary novelty of our hearing actually derived. And this evolution of those auditory ossicles are, again, another example of how evolution is not goal oriented. So with chapter 23, these broad patterns of evolution, I highly suggest you read that chapter. Make sure you do practice problems in calculating half-life. Once you do about 3 or 4 of them, you'll get it down and all is good. And then just know some of those major terms and just kind of these big, broad ideas. Again, not a lot of detail in this particular chapter. So once you're done with that, make sure you guys do your practice FRQs and then also ask me any questions that you have on it. But anyway, thanks for listening. Hopefully this helps clear things up and I will talk to you guys soon. Let's get you out of here and on to, come on. Oh, Mrs. Shelton, come on, figure this out. I got it now. We ready? Okay, signing off. See ya.